You can log them into the Q&A tab. So without further ado, I'll hand this over to Dean Bass.
Yeah, hello everyone. Thank you for being with us. It really is a great honor for me to introduce our upcoming speaker, a most distinguished alumni of the George Washington University School of Medicine and Health Sciences. He's here to present the 9th annual Frank Miller Lectureship. For those who don't know Dr. Miller, he is really, truly one of those most beloved of faculty members ever at the School of Medicine and Health Sciences, and was an anatomist, a pathologist, and won everything in the book that you can win as a teacher and educator, and one who really contributed greatly to our institution through that legacy. We're really glad today that we have his daughter, Catherine Sluter, with us today. She has worked in our library for many years, and she does all of our technology access and serves our students herself in an incredibly effective and important way in our campus. So thank you very much for being here with us today, Catherine. I hope, I hope your family, hope this kind of event helps your family recognize the great contributions that you and your dad made to this institution. Our speaker today is Dr. Richard Whitley. Dr. Whitley currently serves as a distinguished professor of pediatrics, a professor of microbiology, medicine, and neurosurgery, and vice chair of the Department of Pediatrics at the University of Alabama at Birmingham. Really one of the fabulous research institutions in our nation. He's a 1971 alum of the George Washington University School of Medicine and Health Sciences. And since after his graduation, he went on and trained in peds and pediatric infectious disease and virology at UAB, and then went on to an absolutely incredible career as a clinician scientist focused on development or identification and development of viral illnesses in children and adults. But one of his things over this glorious career as a clinician scientist, much of it spent at the NIH, the NIAID, was the evaluation and risk for emerging viral threats. Couldn't be more timely than what we're dealing with today with SARS-CoV-2. He was instrumental in the development or the nation's response to the H1N1 pandemic, and served on a very small panel group that really both looked at the national response to that important pandemic, which was met successfully. And of course, along the way, he somehow or the other got involved in the development of novel therapeutics, including remdesivir, which I hope is the story we're going to hear today, and I believe it is. He's been recognized with numerous national awards for his remarkable record of scholarship and translational science, and including one including the inaugural recipient of the Distinguished Clinical Research Scholar and Educator in Residence at the NIH Clinical Center. He's also received the Alexander Fleming Award for Lifetime Achievement from the Infectious Diseases Societies. He served in numerous leadership roles in every notable infectious disease organization of note. So clearly we are going to hear from a remarkable clinician, scientist, innovator today. So Dr. Whitley, thank you so much for being with us, and I really look forward to your presentation, and I know I'm going to learn a lot. Thank you for being here.
Thank you so much, Dean Bass. I'm going to share my screen, and I'm going to pull up my slides, and we'll put them on full screen. And there we go. First of all, I want to thank the School of Medicine and the organizers for the opportunity to be with you this afternoon and to deliver this Frank Miller lecture. I'm going to say a little bit more about Dr. Miller in a minute, but I want to get my disclosures out of the way. And the first one is important because I am on the board of directors of Gilead Sciences, and I am going to talk about remdesivir, but I can assure you that I will talk about the Achilles heel of remdesivir and where we need to go in the field, as well as anything else. My grant support is only from the National Institutes of Health, and we'll talk a little bit about that. And I will discuss non-approved therapeutics. So Dr. Miller was a fascinating professor for me. Not only was he a professor of pathology par excellence, but he was a Renaissance man, as I'm sure his daughter Catherine knew. He taught an elective that I was really particularly pleased to take, and it was called Shakespeare in Medicine. And at the time, I had no idea that I was going to spend a lot of my life studying herpes simplex virus infections. But I'll never forget the quote from Romeo and Juliet, Act 1, and it was from Queen Mab, the midwife of the fairies, or ladies' lips, which of kisses dream, and off the angry mob with blisters plague, because their breasts with sweetmeats tainted are. It's something I'll never forget about Dr. Miller, truly a remarkable man. So I do want to just bring us into focus now about emerging infections. Emerging infections are not going to go away. They're going to be with us. We know that deforestation, climate change, transportation, all will impact on the world experiencing increasingly new and emerging infections. So I'm going to talk a little bit about the work we do in AD3C, which is the Antiviral Drug Discovery and Development Center. It's a group of people who have aligned themselves to try and develop drugs for emerging infections. The key people are not me, but the three ladies on the right, Mikey Evert, Stephanie Moore, and Mary Wyatt Bowers, who really drive the program from behind the scenes. The scientists do the work, but if they don't sort out the money and the details with the National Institutes of Health, we're in trouble. So this grant is set up, and if you'll just look at the column on the right, where there are four major projects. One is with coronaviruses, led by Mark Dennison and Ralph Barrick at Vanderbilt and Chapel Hill, respectively. Project 2 is alpha viruses, which is Venezuelan equine encephalitis, Eastern equine encephalitis, and more importantly, chikungunya. And that involves Dan Streblow at Oregon Health Science University, Mark Heisey at UNC, Tim Morrison at the University of Colorado, Denver. Flaviviruses with Pei-Yung Shi at University of Texas Medical Branch, Jay Nelson and Alec Hirsch, again at Oregon Health Science University, Mike Diamond, and then Project Floor is influenza that I'm responsible for. In addition, we have a screening core, which uses high throughput technology to try and identify molecules that are active against these viruses. In addition, and I'll go back, we have a medicinal chemistry core, which is essential to the work that we do. This is what I call team science. No one individual can do the work that is done by this group of people. So it takes all of us working together to try and solve these problems. And I just point out, if you look at the four projects at the bottom, coronaviruses are a key player in what we're attempting to do. But even more importantly, with the other three projects, we have proof of principle data for new molecules that we will put in the NIH pipeline, helping develop drugs for chikungunya, all four serotypes of dengue, as well as Zika and West Nile virus. Okay, with that in mind, I'll just point out that the sites that are involved range from Oregon to California to those in the South and the Southeast. And as I mentioned, it involves not only scientists, but it also involves Gilead Sciences, which was a pharmaceutical partner for what we're doing, and provides evidence of the success that public-private partnerships can have. So let's talk a little bit about coronaviruses. Coronaviruses are single-stranded RNA viruses that have a crown-like structure. They're the second most prevalent cause of the common cold. They like to live in the upper respiratory tract, particularly the nose and the throat. They're easily transmitted by respiratory droplets in direct contact. And we know the control of transmission is difficult. It's a lesson that we should have applied to SARS-CoV-2 and COVID-19. And we'll talk a little bit more about SARS and MERS as well. Coronaviruses evolve mainly from lower animal species. In particular, they evolve from bats and rodents. They're transmitted to intermediary hosts, which can be camels, cows, or pigs, and then ultimately transmitted to humans. And we now know that there are numerous coronaviruses that can infect human beings, but most of them are relatively benign. For human endemic coronaviruses, the ones that we most commonly see and are identified clinically are identified on the top line. OC43 and HKU1 are the ones that we'll see in clinical practice and are the common causes of the cold. 15 to 30% of all colds are caused by coronaviruses. There is no durable immunity, so there are frequent cycles of infection. That becomes particularly important when we talk about vaccines directed against SARS-CoV-2. It's going to be difficult, and we need to take unique and different approaches to vaccination and the potentiation of vaccines that will help provide long-lasting immunity. Upper respiratory infections are common. Lower respiratory infections only occur in the immunocompromised host. And we know there's worldwide circulation with winter and spring activity and no licensed vaccines for the common endemic coronaviruses. So the three that we worry about the most are SARS-CoV, which appeared in the country of China and Hong Kong in 2002 and 2004, which is the cause of severe acute respiratory syndrome, causing disease in over 8,000 cases. And you'll remember it began at the Hotel Metropole. A visitor from China stayed there and infected numerous visitors and guests on the same floor. The mortality is 10%. It ultimately involved 32 countries and only existed over a period of 3 months. On the other hand, Mideastern respiratory syndrome, or MERS, occurred in 2010, caused about 2,500 cases of disease, but it had a significantly higher mortality than that which was accounted for with SARS. SARS-CoV came around in 2019, greater than 100,000 cases. Well, that's grossly underestimated now. In the United States alone, we've seen over 1 million cases, and there are over 4 million cases that have been reported globally, and we'll come back to that in a minute, with a 1 to 2% mortality. Bats are the origin, but the intermediary host is probably pangolins, which is a delicacy in China and was also in the fish market in Wuhan. I call COVID-19 the plague of the 21st century. And we're going to spend the rest of this time talking a little bit about what we know of COVID-19 and how we can approach it from medical perspectives. First of all, it behaves like other coronaviruses. Namely, there's respiratory droplet spread, particularly among close contact. That's why we say stand 6 feet apart and make sure that you stay separated for prolonged periods of time. Social distancing, covering coughs, making sure you have a mask, and washing your hands are all critical to the prevention of transmission of SARS-CoV-2. Please remember that there is transmission of virus in blood and direct contact with serum and sputum for healthcare providers. SARS-CoV-2 has been detected in the stool, but we don't at all know what the clinical meaning of that is. Now, I just want to illustrate the number of tests that have been done globally. Earlier this year, September 2nd, 2020, we know that over 555 million tests were done in New Zealand. Now, that's not all in single individuals, obviously. New Zealand tested people again and again and again and again to make sure that they could do contact tracing and patient isolation. Now, just look at the United States. At the end of August, we'd only done 14 million tests. Compared to the rest of the world, that's a paltry number. Let's look at mortality. The United States is leading the world in mortality, although Brazil has caught up with us. When this slide was made, the mortality was greater than 180,000. As of today, earlier, on the Johns Hopkins website, the mortality is over 220,000 people, with an estimated mortality of about 400,000 by the end of the calendar year. Hopefully, containment efforts will do a better job at suppressing this infection. Mortality in the United States has taken an interesting turn. Back in June and July was when we saw the peak evidence of mortality in this country, and it has gradually ebbed. But we know that with holidays and 2 weeks later, namely the 4th of July and Labor Day, there has been a resurgence in cases in many states that have totally avoided social distancing and wearing of masks. And we're seeing that now in Nebraska and Kansas, where the medical intensive care units are at maximum occupancy and the healthcare system is stressed to the extent that it will be difficult to provide additional care. Okay, so what do we know about COVID-19 disease severity? About 80% of cases are mild symptoms. About 14% have severe symptoms. About 5% of individuals are critically ill. With those having comorbid conditions, the mortality ranges between 2 and 12%, generally toward the higher percent. And then with no comorbid conditions, around 1%. The 1% we see actually in young adults, as I'll get to in a minute. So what are the symptoms? Fever is common, 83 to 98%, and that's why taking fever, coming into buildings, going into airports and planes, even going into hospitals in the workplace is so common. But the data here would tell you that we could miss as many as 20% of patients who don't have fever but are infected. Cough is routine, and probably characteristic of this disease is loss of smell or taste, anosmia and ageusia in approximately 30% of individuals. Myalgia and fatigue is common, shortness of breath. For reasons that we don't understand, diarrhea will occur in these patients, and we do know that symptoms will get significantly worse during the second week. It's one of the reasons why early administration of antiviral drugs, and I'll come to that in a minute, will be exceedingly important. Anywhere between 17 and 29% of patients developed ARDS, but with current management techniques, this percentage has come down increasingly, so it's only approximately 10% at the present time. Radiologists think that they can diagnose this infection just by looking at a chest X-ray. They'd prefer having a chest CT, but with a chest X-ray, these lingular infiltrates in the lung are characteristic of disease. With hospitalization, we know the probability is greater in individuals over 75 years of age and even those greater than 65 years of age. Less likely are those individuals in the 18 to 50-year-old age range, and least likely are individuals less than 17. Nevertheless, we know that these individuals are infectious, they do excrete large quantity of viruses, and they are sources of infection for older individuals. And you can imagine the problem this plays as we try and develop thoughtful policies on opening the public schools of our country. Dean Bass mentioned this, but there's a disparity in the number of individuals who get this disease. Underrepresented minorities suffer the greatest number of cases and have both more severe disease and a higher mortality than Caucasians or even non-Hispanic Asians or Pacific Islanders. So we have to address this not only in terms of critical care, but also in terms of vaccine deployment. And the example that I would use, if we don't recruit at least 20% underrepresented minorities in our vaccine studies, it will be very difficult to make judgments about efficacy of any one of the vaccines that are currently in clinical trials in those populations. Dean Bass mentioned Tony Fauci coming yearly to visit GW School of Medicine. This is Dr. Fauci when he was a basketball player in his Brooklyn high school and was the point guard. He was a bulldog then, he's a bulldog now, and he's leading the scientific charge in this country. And I, for one, can say that there's no one better in this country to lead this charge. He's a terrific individual. I've spent a lot of time working with him, and I would do anything for him. So he and Harvey Feinberg, who was then the president of the National Academy of Medicine, previously the dean of Harvard School of Public Health, and he's now back at the School of Harvard School of Public Health, came up with guidelines to crush the curve. One was establish a unified command. And I'd like each of you who are here this afternoon to think about whether we as a country have met these guidelines. Unified command, millions of diagnostic tests available and quickly, supply healthcare workers with PPE and equipment, equip hospitals for the care of these individuals, differentiate the population, infected, presumed to be infected, exposed, neither infected nor exposed or immune, inspire and mobilize the public, and lastly, learn while we're doing real-time fundamental research. Did we succeed? Well, we failed, and we failed miserably. So where do we go from here and now? First, we need to recommend that Tony's recommendations are common sense, and I'll just come to them in a minute. Hospital management requires therapeutics in dealing with this multi-inflammatory syndrome in children, and it was recently reported yesterday in Lancet that a similar syndrome appears to be occurring in adults. We need to address the societal issues of reopening schools, and that will be dependent upon the incidence of disease in the community. And then there's obviously Operation Warp Speed, and I'll make brief comments about that at the end of this talk. Tony's rules are simple. Wear a face mask, stay 6 feet apart, wash your hands. If you're sick, stay at home, don't go to work. If we did this, we would significantly flatten this curve with protective measures. But you can see the states that have failed to do this and what the repercussions have been. We're seeing that now in Nebraska and Kansas. We saw it in Florida and Georgia. We saw it in Texas. We need to just remember to take care of ourselves. If we take care of ourselves and wear a mask, we're not only protecting ourselves, but we're protecting the people around us. So let's talk about small molecule development. And I'm going to talk about 2 drugs and the differences between the 2 of them, and why one has an Achilles heel that we need to think about, and where we need to go from a therapeutic perspective in the future. Coronavirus antiviral research is a team effort. Our work through 83C involved Vanderbilt, UNC, Gilead, Emory, UAB, and certainly individuals at the NIH who helped us with this Center of Excellence in Translational Research. I mentioned earlier that it's a public-private partnership. If it was not a public-private partnership, I don't think we'd have a drug available for us today. With Emory, we have a second drug that's now in clinical trials that I'll make reference to. So what's the story with remdesivir? And you'll note today that remdesivir is broadly available. It's no longer under governmental control. It can easily be acquired through your local pharmacy. It was synthesized by Gilead Sciences to treat respiratory syncytial virus. It was found to be a broad-spectrum antiviral drug with activity against many viruses, including Ebola, coronaviruses, Marburg, and respiratory syncytial virus. We know it failed in the treatment of Ebola in the Democratic Republic of the Congo, but we brought it into the Center of Excellence for Translational Research for reasons that I'll show you in just a minute. We've proven that it has activity in cell culture and animal models, not only with SARS and MERS, but also with SARS-CoV-2. The drug was taken into clinical trials in the United States and China. In the United States, these studies were funded by NIAID. It's the studies that were done by NIAID that led to the emergency use authorization in April 30th of 2020. And it has been amended recently, not only to include just hospitalized patients who are critically ill, but now hospitalized patients who simply have an oxygen requirement. The initial Gilead stockpile was provided to the government free. The drug failed in Ebola. The drugs that were shown to be effective in Ebola were the monoclonal antibody combinations that decreased mortality. We don't know whether it was the dosage of drug, the distribution, or the fact that it's got to be given intravenously that led to its failure. But as we look at the spectrum of activity, the green, darker green, indicates where remdesivir is active against MERS on the right and SARS on the left. And if we just look at SARS, you can see that it's more active against MERS than SARS. in the original cell culture system. But if you look at MERS, we have other studies which would indicate that remdesivir is more active in SARS than it is in MERS. Just because it doesn't have great activity in the initial screens doesn't mean it would not have activity in animal models. And so we started with antiviral cell culture data work. And I don't want to bore people with laboratory experiments, but you do need to know what the IND was founded upon. Here is very simply uninfected cells, cells treated with remdesivir, cells treated with virus. These are decreasing concentrations of remdesivir. And in the top, you can see the progressive destruction of nuclei with virus. You can see as the concentration of remdesivir decreases, there is an increased evidence of pathogenicity. Now we can take these data into more sophisticated animal studies and do what we classically call inhibitory concentrations of drug with cytotoxicity assays, and then combine inhibition of cells with inhibition of virus to show that these drugs are in fact active in cell culture. And it's illustrated here where the drug is very active, as shown by the blue line. Okay. I am going to have to get these slides to move forward, backward, or around. Maybe I can get Rose to help me advance the slide. My slide advancing decided it didn't want to cooperate. There we go. So what happens when we use remdesivir in an animal model that actually looks at lung hemorrhage? And when we do that, 4 days post-infection, we can show that there's little difference between vehicle and drug, but 6 days post-infection, virus is replicating wildly with an increased hemorrhagic score in the lung, and it's significantly decreased with the administration of remdesivir. The drugs thought to be originally useful were Kaletra and interferon, and those drugs had little effect 6 days after the onset of infection when compared to the original drug itself. And the actual hemorrhage is shown in the panel on the far right. If we look at what happens with pulmonary function in a mouse, these are great studies to perform, we show that you can improve pulmonary function with drug compared to the administration of Kaletra or interferon, which had no effect whatsoever. So as we go on and begin to think about how we use these drugs, I just want to mention there's another drug that's in development that you'll be seeing shortly. It has a very simple name. It's NHC. It's synthesized by the Emory Institute of Drug Discovery by George Painter and Greg Blooming. It's currently in phase 2 clinical trials, and it solves an important problem. It's orally bioavailable. And if we look at the basic science data for this drug, we can show that it has a significant effect on decreasing lung hemorrhage, decreases the concentration of virus in the lung, and improves pulmonary function. It does mutagenize the viral genome. So this is not a drug that we would want to administer long-term because it will have significant toxicity. So where are we then with antiviral activity of these two drugs? NHC or EIDD 2801 and remdesivir protect against weight loss, hemorrhage, pulmonary disease when administered 12 hours after infection and after 24 hours after infection with SARS. 2801 and remdesivir diminished lung titer in the treatment groups, again, when administered 24 and 48 hours after infection. These were data that were very important, allowing the Food and Drug Administration to issue the investigational new drug for the National Institute of Allergy and Infectious Diseases studies. So what are our interventions in October of 2020? There's an emergency use authorization that allows the use of remdesivir. NHC is currently in phase 2 clinical studies, And the big difference between the two is NHC ultimately will be given as an outpatient for individuals who are infected before disease is severe. Remdesivir will continue to be given intravenously, although an aerosolized formulation is being developed. Plasma from recovered patients is being studied, as is occurring at George Washington University School of Medicine. Modulators of inflammatory response are under evaluation. They include a variety of different approaches, mainly JAK2 inhibitors, but also corticosteroids. Corticosteroids are available on an emergency use authorization protocol, theoretically for hospitalized patients, but they've even been given on an outpatient basis to individuals who have a simple oxygen requirement. Well, what does this drug do? What does remdesivir do clinically? It decreases the duration of hospitalization by 4 days. It accelerates the resolution of illness. Yes, it will decrease mortality, but this is not peer-reviewed. So the EUA does not include reduction of mortality. Dexamethasone, theoretically, is only for ventilated patients, but it is used in many other patients as well. It will decrease mortality from 50% to 20%. Its combination with remdesivir is not being used in a controlled setting, but it will probably result in even a further decrease in mortality to below 7%. The monoclonal antibodies that are currently being studied as inhibitors of inflammatory response are only in ventilated patients and again, decrease mortality, but are under peer review. Vaccines are in development and we cannot ignore them. There are a variety of different approaches that are being taken. There are DNA vaccines, viral vectored vaccines, subunit vaccines, virus-like particles, inactivated virus, live attenuated viruses, and naked DNA. And I just want to list some of the vaccines that are in progress. Dean Bass mentioned the Moderna vaccine, which is under evaluation at your own institution. That's terrific. They will recruit 30,000 volunteers. This is an mRNA vaccine, which is very, very different than the vaccines that have been used in the past. The only other mRNA vaccine is one that is being developed by Pfizer, and they intend to study 60,000 volunteers. AstraZeneca has a chimpanzee adenovirus vectored vaccine of the spike protein. Novavax has a SARS-CoV-2 spike protein with a matrix adjuvant, and Johnson & Johnson and Janssen are using an adenovirus vector. These are not small vaccine studies. And I want to emphasize that Operation Warp Speed is encouraging, if not demanding, that those vaccine trials funded by the government, namely Moderna, AstraZeneca, Novavax, and Johnson & Johnson, recruit at least 20% of minority individuals and also individuals over the age of 65. Pfizer's vaccine studies and clinical trials are not being supported by the government, so they're basically adhering to these rules, but operate under a different system. There is one data safety and monitoring board for the NIAID vaccine studies, 4 clinicians, 3 ethicists, 3 biostatisticians. Their charter is to monitor all vaccine studies supported with government monies. As I've already mentioned, only Pfizer has opted out. And they have the opportunity to examine the human response across all trials. And the purpose of this is to accelerate vaccine licensure as quickly as possible. I'm going to stop here, but I want you to have my email address. In case questions come up after this talk, I'd be glad to answer any of them for you any way possible. And this is where I live. It's Children's of Alabama. It's a wonderful facility, and I'm happy to be there. I'll be glad to entertain any questions you might have.
Well, thank you so much for that fabulous presentation. Really, what a wonderful body of work, and such a timely discussion of what is right here front and center for us. I guess I would have, and I would encourage the group, if any of you have questions, to please chime in on the Q&A function, and I'll try and read them, and we can go from there. I guess one of the most fascinating things about this right now to me has been this public-private partnership that has really propelled, I suspect has really increased the pace at which this work has been done. I wonder if you could tell us about how did that get launched so effectively? What are the incentives in it? And in the end, what should the financial distribution, return on investment sort of strategy that comes out of these kind of partnerships be from the
That's
institutional?
It's a terrific question. The public-private partnership with Gilead was built upon trust between myself and the chief scientific officer there. And the idea behind it was, just as you said, to accelerate drug development. We had the animal models of MERS and SARS. We also had the capability of doing the cell culture work that Gilead did not have the capability of doing. So we could take those molecules and put them into our systems. What I did not mention, but what is key to the work, is also trying to determine where this virus will develop resistance to the drugs that we're administering. I don't see this as a disease that we're going to end up treating with one drug. We're going to need to treat it with 2 drugs. We're going to need to treat it on an outpatient basis, and we're going to need to treat it as quickly and as easily as possible. And the last part, the distribution of funds, is there's an adjudication committee between the government and the company as to who's going to share in the profits and what's going to be done. Gilead did it as a public health measure. They probably have led the world in what is known as the access program, where they have basically given away their drugs in the developing world. And they view developing a drug for COVID-19 in the same fashion as giving away their HIV drugs in sub-Saharan Africa.
Thank you very much. Okay, here's a few questions we have here. Actually, you did mention what the Achilles heel of remdesivir, and there's a request to expand on that. Is it simply delivery?
Sure, it's simply delivery. The drug has to be given intravenously, so it makes it difficult to be given on an outpatient basis. Sure, we could put a PICC line in and bring people in every day, but then you run the risk of transmission, and you run the risk of facilities and contamination, and that's not ideal. What we really need to do is develop an outpatient therapy that we can give very, very early in the course of disease, because we don't have the biomarkers yet to predict who's going to go on. Sure, we can say people who are morbidly obese, people who are hypertensive, people who are immunocompromised, underrepresented minorities, they're all at risk and we need to treat them right away. But we don't know which of the 18 to 55-year-olds are at risk for progressing. And there are a few that will die in that group, and we need to treat them. And if we don't need to, if we don't know the biomarker, we may still need to treat them because of the complications we're seeing in those patients long-term. Long-term follow-up is going to be essential for everyone with COVID-19 infections.
Yeah, thank you. There's also a question about, let's presume there is a vaccine or even a novel therapeutic that really makes a difference. There's questions about the concern of acceptance of these novel therapeutics in communities of color, people of color, and other underrepresented minorities, given the historical misuse of such therapies at times for those populations. Can you speak to that?
Oh, yeah. I mean, that is an issue that we address very frequently in all the vaccine studies and treatment studies that are being done. There is an essential lack of trust on the part of underrepresented minorities in participating in these studies. It takes a significant effort from community representatives who have the trust of the people who live in those communities to recruit volunteers to come into these studies. I've learned a lot from a woman on the faculty at Johns Hopkins by the name of Lisa Cooper, who's provided strategies for recruiting underrepresented minorities. And the COVID Prevention Network that's sponsored by NIAID is now hiring community representatives to enhance enrollment and to try and build trust. I hope it works. I don't know. We'll have to see. The proof of the pudding will be in the eating.
Yeah, absolutely. We've been fortunate here in our Moderna trial, a number of our very visible Black leaders in the DC government, for example, have been participating in the Moderna trial. And the Washington Post, our press, has been a great ally in elevating that visibility of their participation on a repeated basis, which our mayor and, of course, our health service hopes will facilitate acceptance of the vaccine when and if it's ready to go in the months and years ahead. There's a question here, another one here. It says, given your experience from Dr. Deaton, who you may know.
I know him well.
Yes, of course you do.
It goes back to the AIDS Data Safety and Monitoring Boards of the 80s.
Exactly. And Dr. Fauci, of course, a common thread between the two of you as well. So I would say his question is, given your deep experience with HIV, what are the lessons you learned from that as applied to where we are with COVID-19 right now?
So, I actually was going to elaborate on your statement about the Moderna studies in Washington, D.C. that fits right in with Bopper's question. I think GW as an institution has been able to build trust in the DC community through the studies that were done with HIV and then with HCV. And those studies, a lot of them were, promulgated by folks at the NIH who are on your faculty, like Henry Mazur, who wanted to work with your faculty to make sure drugs were available in the community. And they were accepted by underrepresented minorities. Great success, great accomplishments in the community. So to go back to Bopper's question, keep doing what you're doing because you're doing it right.
Thank you. Okay. Let me ask, I know that I'm sure that your groups as well have speculated about who to best distribute these effective therapies and vaccines to when they're available as well. Can you talk a little bit about that hierarchy and what are you thinking about that?
I think we need to keep in mind that each of the vaccines that are under study may have a different use in different populations. So one vaccine may be efficacious in the elderly, another one may be efficacious in underrepresented minorities. We don't know yet. But there was a wonderful National Academy of Medicine, National Academy of Science webinar yesterday morning that outlined what the potential high-risk populations are and their thoughts on who should be immunized first. And I would point out that if you go to the National Academy website, the document that's been produced by the academies is there, but they targeted frontline providers as the first people to be immunized. Healthcare providers, EMTs, firemen, policemen, nurses, all of the people who are taking care of the patients who have SARS-CoV-2 infection, COVID-19 disease. And if we don't start there, we're never going to be able to get drugs into the people who really need it. So that's where it's going to begin. And then it would move to populations of greater risk, the elderly, those with comorbid conditions, and then finally to the population at large.
Very good.
And we have to remember that there are no studies in children yet. So what are we going to do about the kids?
Yeah.
And that's what we have to consider.
Okay. Here's a slightly more technical question. And does the mutagenicity of NHC increase the likelihood of the mutations that increase pathogenicity and mortality?
Well, insofar as the mutations in the RNA genome with SARS-CoV-2, when we look at it in cell culture, does not render the virus more pathogenic. In fact, if anything, it renders it less replication competent. But I don't want to assume that that's the case in humans. With NHC, we are seeing a propensity for developing resistance earlier. And we're just going to have to wait and determine whether that occurs in humans. Fortunately, it's going to be a short course of therapy, no more than 10 days, probably 5 days. It's exceedingly active. It may end up being given with remdesivir. It certainly will be given with corticosteroids. And it'll probably be given with a JAK inhibitor. So as we go down this road, we're going to have to see how the combinations work.
Yeah, very good. I just have one last closing question. Having this remarkable career as a clinician scientist that you've had, you know, we always talk about experiments working and about wins in science, right? I mean, we got a real win on this one. How, if you look at that arc of your career, you know, sometimes it's hard to keep your optimism going forward when those wins get hammered down over. Can you tell us a little bit about your philosophy of discovery and translation and what keeps you going?
What keeps me going is the smile at the end of the day when you see a patient whom would have died live and not only do well. And I can say that for neonatal herpes, herpes simplex encephalitis, congenital cytomegalovirus infection. And now when I tell you what we're doing, in addition to developing drugs for emerging infections, you're going to say, what? Are you crazy? One of the things that we've done is we've engineered herpes simplex by taking out the gene that's responsible for replicating in the central nervous system. The virus will replicate in cell culture, in dividing cells, but not in postmitotic cells. And in adults who have recurrent glioblastoma, long-term survival is about 20%. We didn't want to brag about it because it wasn't 50% or 60%, so we just sort of shut up. And we made a new virus, and it's a virus that not only is deleted in that gene, but it expresses human IL-12. And we're seeing significant long-term survival in adults with recurrent GBM. But I think what I'm proudest of is in children with recurrent GBM, we have got a 3-year survival rate greater than 50%. And these are children whose Karnofsky scores are sky high. They're doing well. They have no evidence of recurrent disease. And the tumor bed simply looks like Swiss cheese. So, you know, I can't say enough for it, but I have to say that this couldn't be done without a team of people. I mean, the herpes simplex work is done in collaboration with an investigator at the University of Chicago, and the neurosurgeons at UAB, and a pediatric neuro-oncologist. The antiviral studies have been done at 50 sites around the country and involve parents and families and advocates and making sure we were doing things properly. And you said it before, it's transparency. It's making sure everybody's playing on the same team, doing the same thing, and that families understand that there is a placebo in some of these studies, and we have to live with that. So, I mean, would I trade it for anything? No. This has been a wonderful, wonderful ride.
Yeah, no, it's really fabulous. I think the thing you started out studying as a pathogen is now one of your most potent tools for treatment of some rare. It's really fabulous. It's just fabulous. So thank you for all you've done. Thank you for your commitment and long-standing perseverance, and for your creation of teams that really can do this work. We really appreciate that. And we also appreciate you being here with us today. I hope we'll get to meet in person someday, and I really am grateful for your presence here.
We'll do it.
Thank you.
Good luck with GW and running the School of Medicine. You'll have fun and you'll do a terrific job. I loved your presentation.
Yeah, thank you so much. It's been fun so far, and we got lots to do, and we're going to do it. So thanks so much.
And thanks to everybody who stayed until the bitter end.
Thank you. I agree. Thank you all for being with us today. I hope this concludes our reunion weekend events. Off with out with a super firework kind of fabulous thing. So enjoy the rest of this beautiful weekend wherever you are. Thanks so much.
Thanks.
Thanks for joining us, everybody, and thank you very much, Dean Bass and Dr. Whitley. What an amazing talk that was. I want to thank everybody for joining us for Reunion Weekend. I want to especially thank our wonderful class volunteers who rallied their classmates for an amazing and emotional evening last night. I also want to thank Dean Bass for her State of the School update. And finally, all of this could not have happened without the amazing folks on my team. A big thank you to Rose, Jenny, Paolo, and Drew. We were all disappointed we could not be in person this year, but through technology, we're still able to catch up with old friends. It is our hope that we can be in person next year. Have a wonderful rest of your weekend. Please stay safe and raise high.